Hello, my name is Amelia Truong, and I'm a senior at Holt High School in Wentzville, Missouri. I want to share with you my project analyzing genes relating to reactive oxygen species in Stylophora pistillata corals under short-term thermal stress. Climate change is causing corals to die at a devastating rate. This process is the result of the disruption of the symbiotic relationship between corals and algae, resulting in coral bleaching. It is commonly believed that the overproduction of reactive oxygen species by the coral symbionts is responsible for bleaching, but details of this topic are still under investigation. A better understanding of the progression of ROS production over time in relation to other bleaching factors could benefit future studies of coral bleaching. Short-term thermal stress is reversible for some coral species, which might indicate that the widespread bleaching of late could be reversible, but a more detailed study of this ecological microsystem is needed to understand what measures could be done to stop, slow down, or reverse this process. To undertake this project, I reviewed literature on research in this field and found that the latest projects utilized molecular signatures from MicroArray and next-generation sequencing data to study this phenomenon. As a result, I see that the current understanding of ROS is that it is the trigger of bleaching, and specifically in this study, is a result of increased temperature from 24 degrees Celsius to 34 degrees Celsius over a 10-day period. At the molecular level, this steep increase in temperature leads to a cascade of bleaching-related processes in dinoflagellates in the coral itself. To understand this phenomenon, I wanted to collect raw sequencing data that would allow me to analyze gene expression patterns of genes related to reactive oxygen species in Stylophora pistillata coral under short-term thermal stress. Before I tell you about my analysis, there are several terms that you need to know. Firstly, I've been throwing around the phrase reactive oxygen species, or ROS, but what are they? ROS are highly reactive molecules that can cause cell damage or death if built up in the cell. ROS are the cause of the next term, oxidative stress, which is the term for when ROS build up in a system, which in this case is the coral, and the coral is then unable to detoxify itself and repair the damage caused by the ROS. Oxidoreductase is the enzyme that helps to reduce oxygen into a less harmful compound and mitigates the overproduction of ROS. And then after that, we have log two-fold change, which is the data that I'm working with. This is a quantity that compares gene expression values and can reveal if a gene is up or down regulated. I went to the National Center for Biotechnology Information's Gene Expression Omnibus and found 179 datasets related to coral heat stress. Out of the 179 datasets, 18 describe gene expression using microarray or high-throughput sequencing gene expression studies. I collected 8 datasets that are relevant to my problem statement and performed exploratory analysis. In the exploratory analysis, I found that samples could be separated by temperature levels, so I defined my hypothesis that there are genes that will be up or down regulated in low and high temperature environments for a broad range of different corals. After exploring the datasets, I selected the dataset GSE 47779, taken from the publication Gene Expression Profile of Heat Stress in the Red Sea Coral Stylophora pistillata by Moore Landau et al. In that study, the Stylophora pistillata coral are separated into 4 different groups exposed to 4 different temperatures: 24 degrees Celsius, which is the control, and then the treatment groups 28 degrees Celsius, 32 degrees Celsius, and 40 degrees Celsius, which I will be referring to as 28, 32, and 34 respectively. And from that dataset, I searched for genes that are differentially expressed with a threshold of 0.05 p-value and above 1.2 log2 fold change. Then I annotated for ontology function with UniProt and filtered for genes related to bleaching for each sample group comparison. Using the gene ontology information, I searched for the key terms oxidative, repair, and oxidoreductase. The results show a large increase in the amount of differentially expressed genes from the low to medium treatment temperatures, a small decrease in the amount of differentially expressed genes from the medium to high treatment temperatures, giving me an indicator for when significant change starts to happen in coral bleaching. I conducted t-tests with a threshold p-value of 0.05 to compare the results of each treatment for each filter term for both upregulated and the downregulated datasets. The results supported the observation of a stark contrast between the number of genes differentially expressed from the 28 treatment groups compared to the 32 and 34 treatment groups for all the filter terms of the upregulated genes. When comparing the 32 and 34 treatments, the only filter term that yielded a p-value below 0.05 was oxidative. When looking at the downregulated set of genes, the oxidative term had a significant difference between the 28 and 32 treatment groups, as well as the 32 and 34 treatment groups, but not for the 28 versus the 34 treatment groups, possibly indicating that the oxidative related responses for the treatment groups 28 and 34 were similar to each other and different from treatment 32. None of the other comparisons of downregulated genes displayed a significant p-value. Even though next-generation sequencing provides a greater dynamic range for studying gene expression and allows for more accurate measurements of gene expression changes, microarray data can be analyzed and used more easily due to the lack of annotated coral genomes and gene function. In literature, additional factors like genetic background of corals, prior exposure to short-term thermal stress, additional microbes in the microbiome, and their influence on the algae and corals are mentioned. The study did not consider these additional factors. Additionally, this particular study is only 3 to 5 samples per condition, so it's not as comprehensive as I would like it to be. In the article which produced this dataset, the authors wanted to understand gene pathways associated with heat stress. When I started my analysis, I already had a specific question about the relationship between the coral and algae, so I focused on a different aspect of the overall response to the heat stress. Both this study and the original study found that reactions to heat stress began during the 32 treatment, but the original study found that the reactions intensified significantly with the 34 treatment, while this study showed a slight decrease in reactions. This variation in results could be caused by multiple different factors. Firstly, the original study took into account all genes, while this study only focused on a specific group of genes. Secondly, any genes that could not be annotated with UniProt were omitted from the study. And lastly, analysis for the study was performed using Excel rather than using a more powerful analysis tool such as R. In the future, I would like to use a good reference genome to annotate next-generation sequencing data, so I can do this analysis more in depth and with less possibility for mistakes by not using Excel. Additionally, I would like to incorporate more variation in species and look at genetic data and microbiome data to show relationship between multiple factors for biological validation of my analysis. And lastly, I would like to look into mechanistic studies that look at coral oxidoreductase in response to ROS levels in algae.